Defining the autistic phenotypes is not difficult. Understanding it and bringing real-life data and experiences can bring these autistic phenotypes into power. For today's episode, we will cover inhibitory neurons and the autistic phenotype. You likely know about the excitation and inhibition imbalance. In the autistic phenotype, this push-pull tactic the central nervous system uses. Today's episode is all about those inhibitory neurons. In addition, we've discussed the thalamus, our major hub for sensations. All sensations will hit the thalamus and it will continuously provide information in a feed-forward feedback loop to the rest of the brain. The thalamus, because of its massive role, has a specialized shell evolution has equipped it with. Imagine perceiving every sense coming in. You can't. You don't. And I suspect you wouldn't want to. The thalamus has a special feature that appears to evolve alongside with more complex behavioral species. In humans, this is called the thalamic reticular nucleus, or TRN. Now, because humans are humans and scientists are scientists, you can see this abbreviated slightly different, but it is the reticular nucleus. Don't get this confused, however, with areas of the brainstem that create this so-called reticular formation. These are two different things here. The thalamic reticular nucleus is a shell, a thin layer of inhibitory neurons protecting what enters, what signals enter the thalamus. It's a sensory gate system. Now, hopefully you can understand the connection of schizophrenia and autism, the visualization and these heightened senses that we have. Both schizophrenia and autism have an excitation-inhibition imbalance. The outside world is chaotic, and understanding eye gaze is not complicated. Once you understand how those early eye areas for visualizing are in heightened waves, such as gamma, compared to so-called normal, that are in alpha and beta, and slight transient shifts into gamma, all while being able to inhibit noise from the background, which is theta. Theta helps kind of regulate, it scans the brain and kind of helps regulate these waves. See Earl Miller's lab at MIT for some of that. Today's episode is all about autistic brainwave phenotype and the gamma power synchrony with this EI imbalance and the sensory cognitive disassociation with the autistic phenotype. So let's get into the types of inhibitory neurons and their characteristics and roles. But first, what do the inhibitory neurons do? Where do they go? GABA has three receptor types. GABA-A, which is ionotropic, which is fast. These occur within milliseconds. GABA-B, which are metabotropic. These are slower. These are so-called G-coupled protein receptors. These act within hundreds of milliseconds, so they are slower. And a more rare, but still important receptor type here is GABA-C, which is another ionotropic, and these are for sustaining the inhibition response, that inactivation, more in a tonic-like flow, tonic-like activation. These receptors hyperpolarize neurons and modulate neural activity with hopes of maintaining EI balance. The goal is a balance, but with the autistic phenotype, there is a known imbalance. Now, hard stop here. I said tonic-like. And this is true. This is definitely a thing happening in the background. Now, more recently, we've began to talk about waves, these oscillations and frequencies. And all of these molecules, these modulators, dopamine, serotonin, these excitation-inhibition, etc., norepinephrine, all of these are released in different types of kind of frequencies outside of the alpha, beta, gamma, and so forth. These are called tonic firing, burst firing, and phasic. Now, tonic is more regular, single isolated spikes that are in proportion to signals coming in. It's a steadier state in the background, and this can be used to track changes in reward value, such as those internal calculators. Anytime you expose or experience a stimuli, how does these things kind of shift the delta of these? So you can track the tonic, burst, and phasic along with the beta, gamma, and so forth. The other one is burst firing. Now, it is just exactly what it sounds like it is. They are bursting, high frequency, and rapid succession. They are short clusters of action potentials, and they are stronger with more potent effects. Now, an easy example here is like doing a drug. If you do, like, let's say methamphetamine or something, you're going to get a high bursting action of dopamine. Don't do that though. Phasic is more of a pattern of firing that alternates bursts and pauses of activity. These signals are transient or more so-called moment-to-moment changes. And this is also used to allow those internal calculators to evaluate things that are happening in the environment, like reward or value, conflict, and certain outcomes. More event-related than tonic. These are all very fascinating things and very relevant. Okay, we'll hit on those a little bit as we move through the episode. But remember, we have GABA-A, which are very fast acting. GABA-B, slower acting. Still fast, but slower. And then GABA-C, which is kind of rare. So in the autistic phenotype, these dysfunctions will drive sensory hypersensitivity, cognitive and social deficits, and repetitive behaviors. Things that you understand about the autistic phenotype. But we're going to help explain these so-called phenotypes a little bit better. With the specific neuron type, certain brain regions and receptor interactions, and then just general autistic phenotype problems. Now I want to go back before even the central nervous system is created and maybe go upstream here, and something that we've mentioned before in the 2-part neurolation episodes. And that is a fun Sonic Hedgehog gene and protein. It is called Sonic Hedgehog, not Sonic the Hedgehog. Before the central nervous system is forming, Sonic Hedgehog and its mesodermal cells will signal to the ectoderm to create neuroepithelial. Neuroepithelial is the action by which the central nervous system will kind of come in, kind of be formed here. This is the process of the central nervous system. Sonic Hedgehog is very responsible for differentiation and migration. So remember the differentiation. Cells become specialized cells, and then they migrate to similar cells. They migrate to their area of operation, kind of. See also P10, SHANK3, and fibroblast growth factors, and WNT proteins on this kind of epoch that we're talking about. All of those things that I mentioned are regions of interest for the autistic phenotype during this early embryonic development. Now, I say most of that to say this because this is huge. Sonic Hedgehog is critical for developing inhibition type neurons, and an area that we're going to discuss a little bit in detail later with that thalamic reticular nucleus. Sonic Hedgehog is responsible for progenitor proliferation here that guides in the cortex and the thalamus. And it supports maturation and synaptic integration here. This is huge. For the thalamic reticular nucleus specifically, here it is. Sonic Hedgehog established the ventral domain, and it enforces GABAergic identity, which are patterns. If you knock out Sonic Hedgehog, you will see this in research paper. They'll have two minus signs. It'll say SHH minus minus. The rodent or the living organism, whatever they're studying it, will not develop part of the thalamic reticular nucleus. The Sonic Hedgehog gene and protein is responsible completely for developing the ventral part of this shell. But it has no role with the adult formation, meaning you can't knock it out in an adult model and do anything to this reticular nucleus. It's already established. So for development, this is vital. And if it's impaired, the thalamic reticular nucleus and these inhibitory neurons, two different things there, are going to be impaired or even missing, completely missing. And this is shown with models for epilepsy, the autistic phenotype, and schizophrenia. One additional thing before we get into the specific neuronal types of the inhibition. A former guest, Ezekiel Ben Ari, has discovered that GABA during embryonic and fetal development, during gestation, GABA is excitatory. It's not until the birth that is shifted to inhibitory. There are certain sodium and chloride and potassium, I believe, if I remember correctly, interactions that kind of guide this. You can check out the episode I did with Ezekiel Ben Ari, and in the show notes of that episode are all those papers. He explains it well and uses a PowerPoint for easy understanding. So the first inhibition neuron, we've talked about it. We have a whole episode on it, and it's called Parvalbumin interneurons. These are the most dominant type of inhibition that we have in the brain. These are fast spiking and expresses Parvalbumin for rapid calcium buffering. Brain regions here for the Parvalbumin interneurons are the cerebral cortex, specifically the dorsolateral prefrontal cortex. Remember the spindle neuron episode. We found clusters of spindle neurons. They're very few and hard to pick up in the dorsolateral, those spindle neurons. And also the ventromedial prefrontal cortex. This is a huge region of interest because of its massive connections, bidirectional connections to all the neuromodulatory nuclei here, such as the acetylcholine, serotonin, dopamine, epinephrine, norepinephrine, and so forth. And there's also a lot of connections with the subcortical areas here. This is a big hub here. And also the orbital frontal cortex, primary visual cortex, and the primary somatosensory cortex, primary auditory cortex, so V1, S1, and A1. Huge for sensory processing. In addition, Parvalbumin, which is an area that we covered in the episode about Parvalbumin, the interior insula, which is emotional interoception, understanding what you're doing in the environment, how you're feeling, and also the posterior insula. So the insula is loaded here. And the ACC, anterior cingulate cortex, which is kind of like a similar hub to the ventromedial prefrontal cortex. It's a huge internal calculator here. The superior temporal gyrus, remember the decoding the brain, how reading works in autism and dyslexia. We talked a lot about the superior temporal gyrus. And lastly, the inferior temporal cortex. So within the cerebral cortex alone, Parvalbumin has massive roles. It's also involved with the hippocampus, both CA1 and CA3, and the dentate gyrus, and also the striatum, the caudate imputamen, and also the nucleus accumbens, so both the dorsal and the ventral striatum. We talk a lot about the striatum because of the basal ganglia. It's a major network here. Parvalbumin is, of course, involved with the thalamus, the thalamic reticular nucleus, and the amygdala. You mostly, or probably, know about the amygdala for fear. Well, I have news for you. The days of certain regions responsible for certain states, emotions, feelings, and so forth is over. We're over that. The amygdala, yes, is very involved with fear, but it's also involved with much other different opposing types of emotions or feelings and actions. It's more about neural correlates and pathways and connections and networks instead of certain regions. That's a very bad misnomer, and hopefully that will continue to shift. We'll shift out of that. And also, finally, the cerebellum. We've had an episode about the cerebellum with Reza Shadmir. And the Parvalbumin is helping to modulate Purkinje cells. So the Parvalbumin will hit on all three of those receptors, A, B, and C. It will hit GABA-A to help modulate pyramidal neurons, which is our most dominant excitation type. We'll do a whole episode on that. GABA-B receptors for a slower inhibition for potassium-calcium channel modulation in the prefrontal cortex, hippocampus, and the thalamus. And then finally, and this one will make sense, it hits GABA-C for sustained inhibition in the thalamic reticular nucleus, in the lateral geniculate nucleus, one of the first stops for vision, and the amygdala and cerebellum. And it has a high GABA affinity in these areas. So what roles do these have in typical brains? Parvalbumin drive gamma oscillations for sensory integration and attention and working memory, providing somatic inhibition to prevent hyperexcitability. And this is critical for the TRN sensory gating. So what is happening with the autistic phenotype? The reduced density and function in that dorsolateral prefrontal cortex, the anterior insula, which is mainly what that episode was about, and V1, visual cortex 1, and the TRN impairs gamma oscillations, leading to sensory hypersensitivity. The lack of Parvalbumin here cannot regulate this push-pull system, so it creates that imbalance. And it has massive implications to cognitive integration and social deficits. Last thing for now on the Parvalbumin interneurons. This imbalance contributes to that elevated EI ratio, amplifies this brain-derived neurotrophic factor, excitatory enhancement. Okay, this is studied in Molecular Psychiatry in 2017. Decent paper about this. We'll say a little bit more about brain-derived neurotrophic factor, or BDNF, in the excitation episode, more so. But just to recap, the Parvalbumin interneuron is the most dense, should be the most dense of all the inhibition type neurons, the inactivators. The second most is somatostatin expressing interneuron, SOM for short. These are slower firing and provides feedback inhibition that target the pyramidal neuron dendrites. brain regions here are the cerebral cortex, the dorsolateral, that ventral medial, and the orbital frontal, in addition to the V1, S1, and A1. So those three main sensory processing cortexes as well. Anterior and posterior insula, and the ACC, the superior temporal gyrus, just like the Parvalbumin, but they're also found in the hippocampus, CA1 and CA3 as well, and the amygdala. More so for the social behavior, the basolateral and the cortical amygdala. We haven't really parsed out these different subdivisions of the amygdala too much. Receptors, so it works on all three, GABA-A, and depending on which one, A5 or A1, A2, it's kind of help modulating the tonic, inhibition, or the phasic on the dendrites. It's mostly acting on dendrites here. GABA-B for slow dendritic inhibition, and GABA-C has a minor role in the amygdala and hippocampus for sustained inhibition. The somatostatin helps modulate AMPA and NMDA activity. Those are excitation receptors, the postsynaptic. We talked about those in quite detail in an early episode of the podcast, episode 4 or 5. I can't remember which one, but those are receptors that excitatory neurons will park on. They will connect to. Some general roles here for the somatostatin in so-called typical brains is regulate dendritic excitatory inputs and modulate synaptic plasticity, for instance, long-term potentiation. So we've talked about the hippocampus there. That's something that it does. And it supports, the somatostatins support sensory adaptation. Remember, an overarching theme of the autistic phenotype is a lack of adaptive responses. We talked about the anterior cingulate cortex and the ventral medial prefrontal cortex. These are major regions, major hubs for applying adaptive responses. Remember, the social world is the most unpredictable and most chaotic thing that we do, especially for the autistic phenotype. The role here for the somatostatin in autism, what it looks like for the autistic phenotype is impaired function in the anterior insula, hippocampus, and V1. In visual cortex 1, it reduces tonic inhibition and amplified sensory input that contributes to repetitive behaviors. And this is also linked to the brain-derived neurotrophic factor, which helps drive the excitatory imbalance. So the third inhibitory type of cell here, neuron, is vasoactive intestinal peptide, VIP, interneuron. Some characteristics of this is mediates disinhibition by inhibiting the SOM and the Parvalbumin. So it inhibits inhibitory neurons. Does that make sense? We have disinhibition as well. So it shuts off somatostatin neuron and the Parvalbumin neuron. And this will enhance the pyramidal activity. Now the pyramidal neuron is going to be the major region of interest for the excitatory cells. So we'll have a whole episode on that. But this is going to be huge for sensory integration, sensory processing as well. Some brain regions of the VIP, the cerebral cortex, the same areas. These are all in the same areas. Dorsolateral, ventral medial, visual cortex 1, somatostatin 1, and the auditory cortex 1, the anterior insula, and the ACC, anterior cingulate cortex, and the superior temporal gyrus. and the hippocampus. This time, only CA1, for those that really want to know. And the amygdala, the basolateral again. And this time, we're going to find that the vasoactive intestinal peptides are also in the central amygdala. Now, evolutionarily, the central amygdala is it predates the more lateral areas. It's kind of home to innate type of fear. The receptors here, GABA-A, and this targets the interneurons for disinhibition, like we talked about. GABA-B has a little bit of a role here with indirect modulation, and GABA-C has a minor role again with the amygdala and hippocampus. Another role that it has is expression of VIP receptors, which are G protein coupled, and nicotinic acetylcholine receptors. Now, nicotinic acetylcholine receptors are very, very fascinating. Acetylcholine, you might know, is a neuromodulator. It has two receptors, nicotinic and muscarinic. The nicotinic receptor, just similar as nicotine, right? You know about the nicotine. These are very excitatory. These are very excitatory type of excitation. So the vasoactive intestinal peptide role in so-called typical brains is to facilitate selective circuit activation for attention and sensory processing. We kind of made sense with that, with this inhibition role here, and then how it's interacting with the excitation parts. The role of the autistic phenotype is an altered function in the dorsolateral prefrontal cortex in the interior insula disrupts attention and social processing, which will then, of course, lead to social deficits. And this is a good study in Cell Reports. One of my favorite journals is Cell Press. And this was done in 2019. Okay, so a fourth type of inhibitory neurons. It's kind of a 2-in-1. Calbindin and calretenin, expressing interneurons. So why the 2-in-1? Well, I just assume that these are very rare. They're not real known, not studied that much. But the characteristics here is the calbindin provides dendritic inhibition, which is a pretty common theme here. Calretenin mediates that disinhibition, and it targets those interneurons as well. Brain regions here for the calbindin is pretty similar. Dorsolateral prefrontal cortex, orbital frontal cortex, V1, various layers of S1, layers 4 through 6, so very detailed location here for S1. Calbindin is also in CA1 and CA3 subdivisions of the hippocampus and the dentate gyrus and the amygdala, the basolateral amygdala again, and the cortical amygdala. The calretenin is in the ventral medial prefrontal cortex. So we're kind of separating here, finally. Visual cortex 1 and auditory cortex 1, and also the anterior insula and the ACC, anterior cingulate cortex, and the superior temporal gyrus. There's not a whole lot of information to provide on the receptors, what they're acting on, why they're acting on it. These are not well-known cells here that we're talking about. But the roles in the typical brains is fine-tuning local circuits for sensory and emotional processing and neuroplasticity. The role in the autistic phenotype is shown to be reduced functioning in the interior insula and hippocampus contributes to sensory and emotional deficits. Though this is kind of not well studied. They're just not well known kind of cell types here that we're talking about. But we'll wrap it up with the Purkinje cell, which is well studied, especially in the autistic phenotype. There's a major deficit of Purkinje cells in the autistic phenotype. The whole episode with Dr. Reza Shadmir is so fantastic. He's such a great human being. The characteristics here are, these are GABAergic neurons in the cerebellum only, and they provide primary inhibitory output. It's for precise motor control. Very precise detail here, fine-tuning these Purkinje cells are doing. Brain regions are the cerebellar, the cortex of the cerebellar, and lobules 1 through 5, and they project to deep cerebellar nuclei. Receptors here are GABA-A for fast inhibition into deep nuclei. And then GABA-B for the slower inhibition. Remember, these are slower type G protein coupled. And GABA-C for a sustained inhibition. So more of that helping sustain and tonic flowing of granule cells. And it also helps receive AMPA and NMDA inputs. The role in the typical brain is refined motor coordination and cognitive timing in cerebello-cortical loops. The Purkinje cells here are vastly underrated. One of the most underrated cells in the entire human brain. And if you don't know, the cerebellum is kind of like that miniature brain in the back, the posterior ventral back of the brain. And most species have them. If you look at like a rat or a mouse, it's at the top. But that's because the mouse and rat brain isn't as developed. So anatomically, the cerebellum in like the rat or mouse brain is identical. If you think about the growth and evolution versus the human cortex, it's all structured the same, just less. For the autistic phenotype, the cell loss in vermis, Dr. Shadmir talked about this a lot, and lobules 6 and 7 disrupt motor and social behaviors and contributes to stereotypies. So we covered 6 inhibitory types of neurons there, quite dense in the nomenclature and roles, brain regions, and so forth. So it's pretty dense stuff here, but very important because of the roles of this push-pull tactic system that the central nervous system uses. So let's talk a little bit more about this push-pull system. An EI balance is dynamic interplay between activation and inactivation. We use excitatory, like glutamatergic, and these pyramidal neurons, and inhibitory, the GABAergic that we've just talked about. And this is regulating neural circuitry. The push side, this excitation side, the pyramidal neurons in the cortex and the thalamus drive activity with those AMPA and NMDA receptors. And this is promoting things like learning and promoting sensory and cognitive processing. We must be activated to process things. The pull system, the inhibition, the things that will help keep us in check here, are here to prevent overload, hypersensitivity, hyperexcitability, and ensures precision. This is all about a balance, a healthy movement of a seesaw, or the bumper rails down a bowling alley. This is what excitation inhibition is doing. It's trying to keep everything in check, in line, in optimal ranges. Okay, before we wrap up, I like to think about things from an evolutionary standpoint. Why do we have the things that we have? And what are they trying to accomplish? What are we supposed to be doing with them? So the Parvalbumin interneuron. This is mostly responsible to drive gamma oscillations for sensory and cognitive synchronization. and it gates sensory inputs for that thalamic reticular nucleus, especially in the visual, somatosensory, and the auditory sectors. We'll parse that out in a different episode now. It's too much to add in the thalamic reticular nucleus for this episode. It's just too much. And it also provides somatic inhibition in the cortex and hippocampus. So this is the most dense interneuron, inhibitory interneuron, I should say. The somatostatin interneurons is mostly responsible to regulate dendritic inputs. So it kind of regulates where cells park on dendrites and modulates plasticity and supports sensory adaptation. This is a big thing that humans do, is adapting. But specifically in the cortex and the hippocampus. The VIP interneurons, VIP, this facilitates that disinhibition for helping to select attention in the cortex. Signal and noise is how neuroscience looks at this. Signal to noise ratio. What can you quiet? The calbindin and calretenin, these are more for the fine-tuning of local circuits for the sensory and emotional processing. Purkinje cells refine cerebellar, motor, and cognitive output. Remember, this is all about precision control here, especially with reward, Shadmir said. They're mostly involved with highly rewarding things. So some evolutionary aspects. In early vertebrates, GABAergic interneurons, the Parvalbumin and Somatostatin, emerged to stabilize neural circuits and prevent seizures even. Now you know about autism and epilepsy, for sure. This was reviewed in Nature Reviews Neuroscience. Mammalian expansion. Should be increased Parvalbumin interneuron density in the thalamic reticular nucleus and the cortex support complex sensory cognitive processing. Everything we're doing in the environment is pretty complex. Primate specific increases in the dorsolateral prefrontal cortex and the anterior insula, and this enhances social and cognitive functions. That whole episode was essentially about this. So lastly, if you think about developing, learning to navigate the world, learning your environment, how this is interacting here and interfering. The translation of autism or autistic is autos, A-U-T-O-S, which means self. Do you understand why? The self is much calmer and even a lot more entertaining because of our ability to be present inside. The biology that gives us autism allows us to be comfortable within ourselves. And this promotes accelerated learning as well. This is very fascinating with those visual thinking processes here, which is in large part due to that heightened gamma activity in those early visual processes. The retina ganglion cells, lateral geniculate, and then the superior colliculus and the visual cortex, V1 through V4. These are all gamma waves. These are all heightened in the autistic phenotype. You know about the attention to detail, the inability, let's say, to shift off of something. It's not that difficult to understand, is it? It's not that difficult to understand the autistic phenotype. The major problem here is environmental insults. People not understanding it, not understanding the autistic phenotype, in large part, I suspect, because of them. We love to confirm our bias. We love to confirm what we think and how others ought to be. And this is a huge problem. I have something exciting. I want to introduce a product unlike any other product available. A highlight is the product from Daylight Computer Company created their product based on these factors. The Daylight computer is completely blue light free. It has no flicker. Short wavelength flicker is extremely harmful for our eyes and downstream biology. Light flicker is constantly turning our central nervous system on and off. Essentially, it is like going to a light switch and repeatedly turning it on and off. The problem is, blue light and LED light does this, and it is so rapid you cannot even perceive this in real time. The Daylight computer is the lowest stimulation and foremost for sensory sensitive users. It is no question that the alternative product, especially when used at night, do not address or consider this in their product. It is so toxic to human biology. Big tech corporations have patents on how their short wavelength implicate the human nervous system. And a bonus, despite Daylight computer not having backlight, it is very functional for outdoor use. And of course, increased sunlight is always preferred. I am happy to offer a discount for the Daylight computer. You can use the code AUTISM for a $50 off discount. Again, use the code AUTISM and the discount code for $50 off. See the link in the show notes to Daylight Computer Company, or just give it a quick search in your internet browser. Use the code AUTISM for $50 off. I would like to mention Chroma, lights designed for humans. Chroma, a Seattle-based innovator founded by ex-NASA and Air Force engineer Michael Shapiro, is on a mission to enhance physical and mental health with purpose-built devices, unlocking peak human health, cognitive function, and performance. Shapiro launched Chroma to restore the natural light lost to screens in indoor living, and delivers faster recovery, sharper minds, and better sleep. Their products hit hard. The Iron Forge speeds muscle repair with red and near-infrared light, while the Skylight mimics sunlight to boost sleep and energy. A standout, the Forge lamp, is a portable gem that fires 660 nanometer and 850 nanometer light to energize mitochondria and heal tissues on the go. Remember the 4 red light chromophores on cytochrome C oxidase, frequently talked about in the podcast. This is why their products are built with military-grade durability that have lasting impact. Chroma's tech fuses precision and power. Gallium titride power supplies, smaller, cooler, and stronger than silicone, provide flicker-free light that's easy on the eyes. High-powered LEDs target key wavelengths for skin, tissue, and cellular health, with smart heat management for the lasting impact. Every design decision Chroma makes serves a purpose: to create devices that are precise, durable, and effective for improving human life. Remember, humans use different wavelengths of light for different functions of life. Remember when I ask, what do you think light is? Chroma designs with our biology in mind, from sleep aids and wound healing to mitochondrial energy, full body lights and blue light blocking glasses. They are US made and Chroma ships globally and accepts FSA, and HSA payments. Use Autism at checkout for a 10% off discount. That's Autism at checkout for a 10% off discount. If you are listening to the podcast, listening to the episode, please feel free to leave a review or rating. In podcasting, reviews, ratings, and downloads are huge, and I very much appreciate your feedback. You can contact me on X at RPS47586. We can discuss anything and everything about autism. I very much appreciate your comments and your interactions on X. You can check out the YouTube page for all the videos, full-length videos, shorts, and clips. You can email me, info.fromthespectrum@gmail.com. Thank you for listening to From the Spectrum podcast.